Welcome, everyone. Thank you for coming.
Hello, welcome.
All right, we're on week 10. We're almost done with this quarter.
Almost there.
Christmas.
Hope you guys are ready for winter break. I definitely am ready.
Feel free to drop how you're feeling in the chat. One word would be nice, at least. How are you doing this week 10?
One word to describe your feelings.
Yeah.
Cry, oh no.
Literally me
Same.
every day.
This quarter feels like death. Relatable.
I'm so over it too.
It's been so long. It's like short, but also long. It's kind of weird to describe.
Brain equals
Brain. That's actually true though.
Unfortunately, I can't see the chat, so. Oh, brain equals Jose's background. Hey, for real. Quarter feels like death. Uh-oh. Yeah, how many fires did we have? Seriously, this quarter. It's ridiculous. Yeah.
Too many to count.
All right, I think we're ready to begin. Welcome. I'm Jose.
And I'm Monica.
And I'm Ariella.
We're your biosci peer tutors. Don't forget to, actually, we don't have any more tutoring sessions, right? I think this is the end, week 10. I might still do one for you guys, Monica as well. So you can join us. And please don't forget to fill out those evaluations when you get a chance. We do appreciate it. We've been looking at all your comments and trying to implement some changes so we can get better every time. These are some of the main things we're going to cover today. Yeah, digestive system, metabolism. Actually, we're not doing reproductive system. We haven't gotten around to it in lecture yet, so I don't think we're going to cover it. And yeah, this is my presentation. I hope it makes you hungry. Hungry for learning, of course. So this is the overview, what our bodies need, the anatomy and the things that are pertinent to what we're going to learn today, how we digest different macromolecules, and the different stages that we use to digest these. So here we've got your macronutrients. The big ones are carbs, fats, and proteins. We can't forget about water and the various vitamins that we consume. You should know things like the breakdown of each one of these bigger molecules into the smaller molecules, the monomers. So things like carbohydrates into monosaccharides, proteins into amino acids, fats into fatty acids. Just make sure you know those. Here's your key organs that we use throughout the digestive system. The liver, stomach, pancreas, all the big ones that we're going to mention today, and give you a little rundown of what each one of these does and how they participate in digestion. Here's your gastrointestinal tract. Essentially, it's known as the food tube, and that's important because it's basically an open system. So we're open to the environment. We consume food, we ingest it, and that's how we take food into the body, and so that way it can be processed. We need propulsion to get things moving along the food tube. We use mechanical digestion to break down the food through the muscular contractions in the stomach and along in the small intestine. We're also moving things along with the muscles. Again, we refer back to the smooth muscles for this type of digestion. So we see a flashback of all the previous concepts we've learned and the different systems that we've talked about in physiology so far. Chemical digestion, we're going to go ahead and use things like stomach acid and various enzymes to break down the food. We absorb the food throughout the GI tract, especially in the small intestine, and we accomplish this through things like microvilli, which we'll be talking about today. And lastly, the food is removed from the body through defecation, and that's how we get rid of waste. So the big four of the digestive system that we've talked about involve the mechanical and chemical breakdown of food. That's the digestion portion. The motility is the movement of this material along this digestive tract. The secretion would be how we release those enzymes into the digestive tract. And absorption, it can occur through active or passive transport of these substances into the lumen of the digestive tract, which then goes into the ECF and makes its way to the capillaries. So we begin with the mouth, right? Everybody's familiar with this one. So when we want to make food easier to digest, what do? We chew it up. And when we chew it up, we're breaking it down using our teeth and our tongue. And we mix this food in our mouth with our saliva. And we have types of enzymes in our mouth that help us break down these bigger molecules. They start the process. So essentially, you begin the process of digestion in your mouth. It's not actually in the stomach, in your mouth. And the big one here is salivary amylase. You're going to see that often. It's used to break down carbohydrates into their simple sugar molecules. We also see other enzymes in the mouth, which include lipase. And like lipase would be the one that breaks down fats. And then we also see lysozyme as well. So here we have how we digest things. We start with mastication. That's the medical term for it, for chewing. And then deglutition. This is a fairly new one for some of us. I know it was for me. And that's how we swallow things. The epiglottis is what prevents the food from going down the trachea. So it just goes down your food tube, known as your esophagus. And you don't want to take food down your trachea because then you'll choke. So then the food is moved down through these muscular contractions known as peristalsis, and it makes its way down the esophagus. And when food is within the digestive tract, within your food tube, so to speak, it's referred to as a bolus. And this food makes its way down from the esophagus through that primary sphincter and into the stomach, where it'll be exposed to that stomach acid. Okay, so a fun fact is that we humans can eat upside down. So if you were to hang somebody upside down and feed them food, once you swallow the food, your peristalsis will take over and push that food in the direction that it would want to go for absorption. There's a vast amount of things that we're going to talk about today, like the pancreas, the different pancreatic juices that help us in digestion, the various enzymes. So if you have any questions along the way, you can just say them towards the end because we're going to eventually cover those things. So once the food makes its way to the stomach, it's exposed to that stomach acid, and which will start the process of digestion. And with the mechanical and the chemical digestion of the stomach, you're breaking down that food into those small particles that are more easily able to be digested. So this is the two types of digestion that the stomach is involved in. We've got those muscular contractions along with the chemical digestion. And the big one that was covered in class is how stomach acid, the hydrochloric acid, activates pepsinogen and converts it to peptin. And this will be able to help break down those proteins. So protein digestion begins in the stomach. Okay? And pepsin works by cleaving those bonds in the amino acid sequence. It cleaves peptide bonds. So these are the various layers of the stomach. Let's know the key anatomy, like things like the fundus and know the pylorus and the rugae. The rugae are particularly important because they involve what you see here on the right. Basically are those folds that contain all the various cells that are involved in digestion. And what's important here is that the stomach itself is a very acidic environment, which would mean that you have a very low pH. And because of this acidic environment, you're going to go ahead and start breaking down that food chemically. Now, you don't want to digest yourself. So you do have to produce mucus. And you build up this mucus layer. It's like a barrier that prevents the stomach from digesting itself. And so this is that barrier that we're trying to establish here. And that pepsinogen, once it's exposed to the hydrochloric acid, becomes pepsin, and it begins that process of the amino acid digestion. And be particularly aware of how the pH ranges from the stomach acid once it starts passing that barrier, and then you make your way to what's considered a normal body pH. So then that way, these substances that are making their way throughout the body, once they're broken down, they need to become absorbed. And then when they're absorbed, they're transported through the body. And everything that we're absorbing through the various cells in our digestive tract make their way to these capillaries and are then moved on to the rest of the body. So these are the secretory cells. This is essentially like an overview, so you can take the time to digest this material on your own. Forgive the pun there, but anyways. So not all of these did we cover in class, but it's nice to have a table like this in case something is referred to, you can just look back at it. So, right before you take your exam, have those kind of materials at hand, so that way you can just kind of refer to that material and you can be primed or ready for this kind of things. You can look straight at something and know, okay, the mucus cells, what's happening there, the parietal cells, what is happening there. But yeah, that will help you be able to parse through the different information that we've covered. So we know that the parietal cells are the ones that produce the hydrochloric acid. So the main process that begins here is the water dissociates, and then we have that very famous ATPase pump. But in this case, it's using potassium and hydrogen ions. And what it does is it moves hydrogen into the lumen. So it exchanges the potassium that's inside the cell, and then it goes ahead and moves that hydrogen ion into the lumen. Carbonic anhydrase is that key enzyme that makes the bicarbonate. It helps facilitate the production of that bicarbonate. And you can look at these chemical equations. If you're versed in that sort of thing, you can understand what I'm demonstrating here. This is essentially the same kind of picture that you'll see for parietal cells if you look on the internet or even on our lecture slides. It's going to be the same thing. You just know the pathways, so where each thing is going and what direction they're flowing. For example, we have the bicarbonate and the chloride ion. They exchange. This is known as that chloride shift. And know that that chloride is entering the stomach lumen, and it's being exchanged for that bicarbonate. And then lastly, that chloride, once it's in the cell itself, it makes its way to the lumen through that chloride channel. And that's where it forms the bicarbonate. The hydrochloric acid. So then these are the accessory organs that we'll be talking about. And the big ones are the liver, the gallbladder, the small intestine, the pancreas. So all of these help to aid in digestion. For example, we've got the liver, which makes the bile, and it sends it to the gallbladder for storage. The gallbladder holds on to that bile, so when it's needed, like when you consume fats, it'll secrete it into the beginning portion of the small intestine, and that's known as the duodenum. And then the pancreas helps to aid in digestion by secreting pancreatic juices. That these enzymes break down like fat molecules and fatty acids into glycerol. Fat molecules like triglycerides into fatty acids and glycerol. Additionally, proteins are broken down into amino acids, and carbohydrates into glucose, and that's the whole basis for digestion, is breaking those big molecules into smaller molecules. So once you've made your way through the stomach and you've had that initial portion of digestion, the chemical and the mechanical, you want to make your way down to the duodenum so you can begin absorption. So this is the gatekeeper here, the pyloric sphincter. It serves as that gateway between the stomach and the small intestine. It allows contents of the stomach to pass into the small intestine, but it also prevents partially digested food and digestive juices from entering the stomach. A big thing that we see here is, for example, when people have stomach ulcers and things like that, it's caused by a bacterium in the pyloric sphincter area known as H. pylori. And then you have to take certain medications to go ahead and treat that. So it is a key participant in the physiology of digestion. Another thing is, so this partially digested food is known as chyme, and it empties from the stomach into the small intestine, and it passes through this pyloric sphincter. So chyme is what's referred to as that partially digested food. And the small intestine is the main site for absorption of all of these molecules that we're going to see. So here we have the small intestine. I went ahead and borrowed what the professor Dr. Lutterschmidt put here on the left side, we see the different involvement of the different pieces of the small intestine. I like to remember the small intestine from the duodenum, jejunum, and ileum by thinking DJ ileum. I don't know if anybody's familiar with like EDM music, but DJI. So then that way I know the, the way that it flows through the body. So we know that it makes it to the duodenum first, then the jejunum, and then the ileum. So most of the absorption of nutrients and minerals and from food takes place in this region in the small intestine. It lies between the stomach and the large intestine, and it receives bile and pancreatic juice from the pancreatic duct in digestion. The duodenum is the first and shortest segment of the small intestine. It receives partially digested food known as chyme from the stomach, and it plays a vital role in the chemical digestion of chyme in preparation for absorption in the small intestine. Mainly in the first half of the jejunum is where the majority of absorption takes place. And this is at about 90% of your nutrient absorption occurs in the jejunum, okay? This involves proteins, carbohydrates, vitamins, and minerals in that region. The ileum is the last section of the small intestine, and it is basically what leads to the large intestine or the colon. And its role is to mainly absorb water, bile salts, and vitamin B12. Now, this is a picture of the small intestine with the various layers. And we know that the inner wall of the small intestine is covered by these numerous folds of mucous membrane called the paicae circularis. So you see those round circular folds there. And the surface of these folds are what contain those tiny projections called the villi and the microvilli. And which further increase the total surface area to enhance absorption. So you want greater surface area, which gives you maximum contact with these nutrients, and that way you can absorb them. The function of the submucosal plexus is to innervate the cells of the epithelial layer and the smooth muscle of the muscularis mucosae. And for the most part, there's a huge percentage of these neurons that are sensory neurons, and they're known as afferent neurons, and they send signals up the pathway to the central nervous system. So here, if we take a closer look at the small intestine, this is where basically we're looking at the crypt is where we're making these stem cells that we're going to use to develop the renewal of the epithelium. And every 3 to 4 days, we're constantly turning this over. There, I've included additional things that might not be necessary for this course, the Paneth cell and the transit amplifying cell. But you do need to know, however, the goblet cell, what the enterocyte is and its functions, and how things transport throughout the body. So here's like an even more zoomed in view to each one of these individual microvilli. So the lining of the small intestine consists of these finger-like projections, okay? And they're referred to as the microvilli. Projections, like I mentioned, increase the surface area of the intestine, and it's the region where the absorption takes place. Water-soluble food molecules such as monosaccharides, amino acids, minerals, and water-soluble vitamins are absorbed through the walls of the villi into the capillaries by diffusion and active transport. Fatty acids and glycerol enter the epithelium of the villus by diffusion, and the epithelial cells, they recombine into lipid droplets, and this is something we're going to talk about later on. And these lipid droplets enter the lymph and the lacteal. And then additionally, lipid-soluble vitamins are also transported this way into the lacteal. So this is going to be the carbohydrate breakdown. This is just a big picture look at it. So what we want to absorb are all these monosaccharides. That's the end goal. So you're going to use various enzymes to break these down into their individual components. So the maltase would be broken down into 2 glucose molecules, whereas sucrose is broken down into a glucose and a fructose by the enzyme sucrase. And then lastly, lactose is broken down into glucose and galactose. So each one of these enzymes sounds like the disaccharide. So maltase, sucrase, and lactase, respectively. So here we've got the carbohydrate absorption, and essentially I like this picture because it shows you how each one of those molecules is going to be transported into the body, into the bloodstream, and the individual enzymes that break them down. And here we have a picture similar to the one that we use in the textbook, and just know each one of these transporters and what molecules they move Like for example, the SGLT is moving sodium along with either glucose or galactose into the lumen, and it does this by co-transport, so they're going in the same direction, they're going together. Whereas fructose is moved by GLUT5 into the lumen, and it does I like to remember GLUT5 because fructose, it kind of has like a pentagon shape, so it can help remember that one. And then lastly, all of these molecules exit out the other side via GLUT2 transporter. So the digestion of and absorption of the proteins, it occurs by hydrochloric acid denaturing these proteins, basically messing up their structure. And additionally, the HCl goes ahead and activates that pepsinogen into peptin, which further leaves bonds in the amino acid sequence. So we've got several pictures here that show that breaking down into the individual amino acids. Pepsin is a protease, meaning that it cleaves bonds in proteins. And here's a chemical diagram that shows a polypeptide broken down into polypeptide fragments. So that's what this peptin is doing. It's breaking down those bigger chains into smaller chains. So the absorption of the amino acids, we have these membrane-bound aminopeptidases in the jejunum, which finish off that protein digestion. Amino acids are absorbed by a co-transport mechanism with sodium ions, and both sodium ion and amino acids combine with a cell-separate protein receptor. There are different receptors for the different groups. In addition, certain amino acids may have their own specific transporter. I recall the professor mentioning choline, that's a specific one. This receptor then conveys both of these molecules to the inside of the cell, and the energy for this transport is derived from the concentration gradient for sodium across the cell membrane. So that very famous sodium-potassium ATPase transporter actively and continuously pumps the sodium ions outwards towards the sodium gradient. And after absorption into an enterocyte, amino acids can have one of two fates. A minority, like particularly glutamate, are oxidized to liberate energy, and the majority are passed on to the portal for circulation. And the movement of sodium into the cell is accompanied by chloride and water movement into the intracellular space and eventually the bloodstream. There may be a small amount of absorption of amino acids as di and tripeptides. These are probably digested within the cell for amino acids. So you see these big chains are cut up into smaller chains. We've got those di and tripeptides with hydronium. They're transported in, whereas amino acids co-transport with sodium ions as well into these cells. And then we have small peptides that are just carried without that co-transport mechanism across the cell. Now, a big organ, an important organ for digestion is the pancreas. It secretes those digestive enzymes into the duodenum. We've got various types of cells here that help make this process happen. For example, the duct cells make that bicarbonate. So when we have that stomach acid, it's going to be very low pH, and which means it's very acidic. So we need things like bicarbonate to go ahead and buffer that stomach acid. So that way, when it enters the stomach, when it enters the small intestine, it's not causing that potential damage. Then we've got the acinar cells. They're secreting those digestive enzymes into the duodenum to help break down that food. And then we've got the isolate of Langerhans. These are the ones that are responsible for that endocrine function of the pancreas, which I believe Monica's going to go over with you. It's going to talk about the metabolism, where we use things like the beta and alpha cells, which are responsible for the secretion of those pancreatic hormones. For example, I'm just going to mention this briefly. Beta cells secrete insulin. You know that because that's a big one for physiology. Okay, and it's a well-characterized hormone that plays an important role in regulating that glucose metabolism. So now cells that produce bicarbonate have high concentrations of carbonic anhydrase. Chloride enters the cells by indirect active transport and leaves the apical side through a CFTR channel. And this chloride then re-enters the cell in exchange for bicarbonate. So if you follow these pathways, if you look at where each direction of the flow of each of the ions and the molecules, then you can begin to understand the importance of these. And there's also these leaky junctions in between the ductal cells, and they allow for pericellular movement of ions and water. So these negative ions in the lumen attract sodium, and then by the pericellular pathway, and they move through this in between cell water, and then of course water follows the flow of ions. So if you've got that sodium going through, then the water's going to go ahead and boil. Now fats are digested. So fats are digested. They're broken down from those big fatty acids into the individual monoglycerides, and what happens is they enter this. It's kind of like a bubble. Let's call it a mycel. It's oftentimes when they refer to mycels, they're referring to like soap. Soap forms these bubbles of like that encapsulate things. So what we're doing is we're trapping these monoglycerides within a little bubble, so that way it can be moved into the cell for absorption. And when it makes its way through, these globules is a good way of mentioning. Yeah, fatty globules. They combine with proteins to form the chylomicrons, and this occurs in the Golgi apparatus. Then those chylomicrons are then pushed to the other side of the cell and enter the lacteal for absorption. And these lacteals are those lymph vessels that go ahead and absorb and carry those chylomicrons away from the intestine. So that's how we absorb fatty acids. Know that things like the lipase are the ones that bind to those fat droplets in the duodenum, and we use these to break down fatty acids into those monoacylglycerols, and then we can transport those to the duodenum and the jejunum. So this is an overview of the absorption of the different macromolecules. So we've got your proteins again, your sugars or your carbohydrates, and then your fatty acids and how they're transported across. Okay, cannot forget about the liver and its important role in digestion. It makes that bile, and then it transports that bile to the gallbladder for storage. We've got it's involved in so much metabolism, from food to drugs, hormones. Basically, the liver has a range of functions. It's very important for detoxing or making sure that something harmful doesn't go ahead and hurt you or cause any negative effects on your body. So you want to take care of your liver. Yes, you can refer to a table like that just to have that broad range of information. So this is just a rundown of all the various hormones. These are the ones we talked about in class. So just go back to your notes and refer to the different hormones and know what each one is and what their key function is. I'd like to include the leptin and ghrelin. I don't recall too much of mention of it, but these are important ones in physiology just to know. And just know where they're secreted and what causes their secretion. I created a worksheet for you guys so you can refer to that has a table. So the answer key's there, and then that would be like a key thing to study because it has the answers right there and readily available for you. So lastly, from the small intestine, we make our way to the large intestine. So we're nearing the end here. So know how things move through that large intestine. And these are the 3 patterns of movement. I went ahead and borrowed this from our lecture because I feel like it's a really good overview of how things move. So we've got the migrating motor complex. It's that series of contractions that start at the stomach and make their way to the large intestine. So it's just like the whole movement of throughout from the start to the large intestine. And then the peristaltic contractions are those muscular contractions that are pushing forward the fluid. They're always going to push it in the direction of basically exiting your body. And segmental contractions are a range of different contractions at they're kind of like pushing the food back and forth, up and down, and they're mixing that food. So then that way it can be easier to digest. This is the different sections of the large intestine. I color-coded this for you so it's easier to follow, but just, I don't know if they would ask an ABCD question on this, maybe put it in order, or just know, for example, the cecum and the appendix that are located in the ascending colon. That might be an important one to know. Transverse colon crosses from one side of the body to the other. It transverses to the other side. Ascending means to go up. Descending means to go down. And at the ending here, we've got that sigmoid colon before we reach the exit point of the colon. And lastly, the last part of digestion, elimination. So this is also referred to as defecation. And this is the final step in digestion. Here we eliminate undigested food content, waste products, And after that food passes through the small intestine, the undigested food makes its way into the colon, where most of the water is reabsorbed here. So in large intestine, most of your water is reabsorbed. So we eliminate solid, semi-solid, or liquid waste from the digestive tract via the anus. And that's pretty much it. There's a range of health conditions that are involved in the digestive tract. So just make sure you take care of what you eat and take care of your body. Everybody here might know somebody that's suffering from one of these ailments, or there's so many more that you could be suffering from just because you don't take care of what you put into your body. So I think that's really important for you to watch and see, especially in high stress times like final food, you know. So I have a series of questions for you. Don't mind answering these questions. Can someone set up a poll? Yeah, perfect. So for the first one, what is the first macronutrient? All right, I think we're ready. Let's take a look at our answers. We got, yeah. All right, for the most part, everyone got that right. It's carbohydrates, you are correct. And where does most absorption of nutrients take place? I know these are kind of easy, right? I feel like for the most part, everyone's gonna get this one right. All right, 3, 2, 1. Let's see how you did. Awesome. Good job. Okay. Now let's look at these questions. What do we think here? It's a little bit more difficult. We're gonna give you 15 more seconds. 5, 4, 3, 2, 1. Good job. And let's go ahead with the next one. What are we thinking? So let's just split here. One answer is more correct than the other. All right, 5, 4, 3, 2, 1. Go with your gut. All right. Y'all did good. So some of you, or most of you, are ready for the exam tomorrow. Good luck. I think this might be the end of my time here with you. So I appreciate you, taking the time to listen to me talk for you for a moment. Feel free to send any questions my way, and I'll hand it off to Monica. Thank you.
Awesome. All right. So I'm going to be talking today about metabolism. If we can move on to the next slide. Okay, so this is the first part of metabolism, lecture 20. So the first thing I'm going to talk about is the integration of the central nervous system and the gastrointestinal tract in metabolism. So there are two pathways that you should keep in mind. So there's the intrinsic pathway, and that's going to be the enteric nervous system that's localized within the GI tract. And then there's the extrinsic pathway, which is controlled by the nervous system. So the first thing that happens is you're going to get a stimulus. So that whether it could be something that you see or you smell. So obviously sight is also important for stimulating appetite. Like imagine like you see a burger that looks really good, that could also cause this cascade of events to occur. in your body. So you could say smell a really delicious burger cooking up, so that'll trigger your sensory receptors in the extrinsic pathway. And then that signal will go to your brain where it's going to integrate. And then it can go to the plexus neurons, which via the efferent signal from the brain. And then that can then go into the muscle or a secretory cell. And then depending on where that signal goes, the muscle will either contract or the secretory cell will secrete, it will cause secretion. And then that's how the response then will be triggered. On the other hand, well, it's also important to keep in mind that the relationship between the brain and the plexus neurons is mediated by the sympathetic and the parasympathetic nervous systems. So that's one extra point to keep in mind. Now, there can also be metabolic outcomes. So that's going to be mediated mostly by pH, stomach stretch, and food localized within the GI tract. So that can cause secretory cells in the intestine or the stomach to release these GI peptides. And then from there, a few different pathways can take place. So the GI peptides can either go into the pancreatic islet cells, and then which can then secrete insulin and glucagon, depending on the conditions. Can go to the brain to initiate hunger or satiety versus via neuropeptide Y, which is a neuropeptide that we will talk about later on. That's very important in mediating hunger. And then finally, it can also go to muscle or secretory cells, which can then, like we said, generate a contraction or secretion response. And then just one extra thing to keep in mind is the intrinsic pathway, which is denoted in red arrows. That's going to be the enteric nervous system that's independent from the central nervous system. So that starts by a signal from the pH of the stomach or stomach stretch receptors, or of course food. And then as you can see from the red arrows, that's going to be the pathway that's going to take place. So it's going to go into the sensory neurons, and then it's going to be integrated via the interneurons, can reach the plexus neurons, and then once again reach the muscle or secretory cells. So the hypothalamus is the brain's main feeding center. So there are two portions of the hypothalamus that mediate the feeding behavior. So one is the lateral hypothalamus, and this is the brain's feeding center. And so this part will contain a very large amount of neuropeptide Y. And neuropeptide Y is very important in regulating feeding behavior. So when you're hungry, neuropeptide Y will be activated in order to signal to the brain that you're hungry so you can go ahead and eat. Versus the ventromedial hypothalamus, that's the brain satiety center. And so that's how you're going to know that you're full. And so you're not going to want to eat anymore because your brain is telling you that you don't need to. And then both of these regions mainly receive input from the cortex, the limbic system, and then various hormones that we will talk about next. So there are two theories for mediating feeding behavior. So one is the glucostatic theory, which poses that short-term regulation of food intake is mediated by blood glucose levels. So if you have high blood glucose levels, it's essentially saying you're not going to want to eat food because you're going to feel satiety, and vice versa. Whereas the lipostatic theory, it posits that you're going to be full when you have high fat storage in your body. And so that mechanism is going to be mediated mostly by adipose cytokines and the hormone leptin, which is the satiety hormone. And it's important to keep in mind that these theories are not mutually exclusive and that both of them do take place simultaneously in order to mediate our feeding behavior. So what are some hormones that drive food intake? So let's just go through this cascade step by step. So like we said, the first thing that happens when we want to eat is a neuropeptide Y signal will reach the brain, or a signal will reach neuropeptide Y. And so that will then, that signal will then go into the hypothalamus. And so depending on if we're full or we're hungry, that signal will either be inhibitory or excitatory. And so some peptides that inhibit neuropeptide Y are CCK, which originates from the duodenum, obistatin, which originates from the stomach, and then insulin, which originates from the pancreas. And insulin is an especially important one because when you eat, your blood glucose levels are going to be high. And so the pancreas is going to secrete insulin in order to tell your cells to take up the glucose. And so that causes a negative feedback loop that inhibits neuropeptide Y and tells the brain that it doesn't need to eat anymore. And that's different from ghrelin and orexins. So ghrelin is a peptide that's released from the stomach, and orexins are neurotransmitters that are released from the hypothalamus, and both of these will activate neuropeptide Y in order to encourage feeding behavior. And so you're getting the hungry signal, you're hungry, the hypothalamus is integrating that signal, the feeding behavior is increased, which will increase fat storage, and then ultimately increase leptin levels. So leptin, like we said, is the satiety hormone. I like to think of it, when I first learned it, someone told me to think about leptin tea. Like I usually like drink tea after meals, like when I'm full. So that's how I remember that leptin is like satiety. So leptin, you don't need to eat anymore. You can have your leptin tea. So if that helps you remember it. And for ghrelin, ghrelin is the hunger hormone, so you want to eat. So think about your stomach growling. That's how I remember it. All right, moving on. Okay, so here's a question. You just ate some delicious Chick-fil-A nuggets. Which of the following would you expect to happen? Think about the wording here. Okay.
Wait, do we get sauce with these nuggets?
What kind of sauce would you like? I usually get the honey mustard when I have Chick-fil-A nuggets. You too,
Mustard?
Ariella? The only way.
No.
Chick-fil-A sauce? Okay, I could do that too, but only for the sandwich for some reason.
There you
I don't know
go.
why,
Classic.
not for the nuggets. All right, let's end the poll. All right, let's reveal the right answer. Next slide. Yes, most of you guys got it. Good job. So next we're going to talk about how we measure the metabolic rate. So metabolic rate is defined as the rate at which the body converts energy input into heat and external output. And so there's a couple different ways that we can measure our metabolic rate. So one is direct calorimetry, and that is where we obtain a direct measurement of the amount of heat generated by the body within a particular structure. So as you can see from the figure on the right, this is usually done by like having an animal sit in this chamber that's insulated. So there's like that exterior structure where we'll insulate the interior like container that the animal is sitting in order to prevent like heat exchange. And then you have a bunch of ice surrounding the exterior chamber and the interior chamber. And so the animal, without moving or anything, using its own metabolic energy, will start to melt that ice. And so at the bottom, we're going to collect the amount of water that has been melted by the ice, and that will tell us how much energy that animal essentially burned. So that's a measure of metabolic activity. The only limitation here is that not all metabolic activity is converted into heat energy. So some of it is converted into like external output or like storage. body storage or fat storage. So it's not a perfect measurement of metabolic rate. Next. And then the other method that we use is indirect calorimetry. And that is where we calculate the heat produced by a living organism through this method called respiratory gas exchange. And so that essentially involves measuring the oxygen consumed and the carbon dioxide produced. And it can be used to measure the respiratory quotient. And I could see this being a test question. So I would encourage you guys to memorize this formula. So the respiratory quotient is the moles of carbon dioxide produced per unit time divided by the moles of oxygen consumed per unit time. And so you can see this here in this figure, like using the chemical formula, we can divide the 6 carbon dioxides by the 6 oxygens to get our respiratory quotients. And that can tell us information about what that person's feeding behavior is. So for example, lipids tend to have higher ratios of oxygen consumed to CO2 produced than carbs. And so a person with a certain respiratory quotient may be consuming more lipids versus someone with a respiratory quotient of 1, who most likely just consumes like carbohydrates. So that can tell us certain information about dietary behavior. So resting metabolic rate is the total number of calories that the body burns at rest. So you're not running, you're not walking. It's just how many calories are you burning just sitting down. So a couple of different ways, conditions through which we measure it. So we measure it under the thermoneutral zone. We measure it when the person is fasting but awake, which differs from the basal metabolic rate that's measured when the person is asleep. And then the metabolic rate, it can be influenced by a number of factors, including age, sex, genetics, the microbiome, and many other factors. And males tend to have a higher metabolic rate than females. So nutrient pools and metabolism. This figure can be kind of intimidating. I remember thinking it's just a lot when I first saw it. But essentially what it's showing is that these different substrates can be used to mediate biochemical processes. So for example, we tend to think about glucose being produced via gluconeogenesis, or like glycogenolysis, where like the glycogen in the liver gets broken down to generate glucose. But it can also be made using proteins. So for example, proteins can be broken down into amino acids. And if you have some excess amino acids floating around, the body can use that as a substrate in gluconeogenesis in order to produce glucose if that is a deficiency. And then on the other hand, if the body has a deficiency in protein, it can actually use its own protein reserve to break down those proteins in order to generate amino acids. And so there are these dynamic methods that the body uses in order to generate glucose, protein, fatty acids, whatever it needs, using many different substrates. One key point to keep in mind here in the middle part with the glucose pool, notice how the brain metabolism arrow is the lowest one. So it's much lower than metabolism that goes to most other tissues. And that's because the brain will be the last thing to receive glucose. So if all else fails and you barely have any glucose, the priority goes to the brain, essentially. So the body will make sure that the glucose goes to the brain before it makes sure it goes to any other body part. So just keep that in mind. So I have here a question for you guys. So which of the following may not be used as a substrate in glucose production? Think about the figure that we went over and the directionality. Okay, we'll end the poll in a few seconds. Okay. What was the answer? Yeah, so free fatty acids. So that's an important directionality to keep in mind is, so glucose can be produced via these other pathways, but you can't, for example, use glycerol or fatty acids to produce glucose. So just keep that in mind. And then the main point I wanted to make with this figure is that, like we said before, many different substrates can feed into these biochemical pathways. So, for example, glycerol can be used as a substrate for glycolysis in order to generate ATP, as well as glycogen, right? So glycogen can be broken down into glucose, and then also feed into glycolysis. And then some amino acids can also modify their structure by getting rid of their amine group in order to feed into glycolysis by going into pyruvate, and so on and so forth. And then lactate, which is the last product of glycolysis, can also be fed again into the glycolytic pathway. And then for the citric acid cycle, some amino acids may be used to be fed into that cycle as well. And then also fatty acids. So that's kind of an important process too, is fatty acids may undergo beta oxidation in order to form acetyl-CoA. And acetyl-CoA is the first substrate in the citric acid cycle. And a byproduct of beta oxidation is ketone bodies, which we will talk about more when we discuss diabetes and starvation. Great, lecture 21, metabolism part 2. So insulin and glucagon are probably the two most important hormones that you will need to learn for your final. So in the fed state, insulin will dominate. And then, so insulin is released from pancreatic beta islet cells in response to high blood glucose. So after you eat a meal, your blood glucose levels are going to be pretty high. And so that'll trigger insulin to be released from your pancreas in order to notify your cells that you just ate a meal. The glucose that you just ate needs to be taken up by the cells. So we tend to think of insulin as the anabolic hormone because it's going to signal to the cell that we have all this energy, we can use it to build up molecules and macromolecules and proteins and such. And then vice versa, in the fasted state, glucagon will dominate. So glucagon will be released by pancreatic alpha islet cells in response to low blood sugar. And we tend to think of glucagon as the catabolic hormone because it'll trigger the breakdown of glucose from the body via all these processes that we went over. So glycogenolysis, which is the breakdown of glycogen into glucose, is one option that the body can do. So glycogen will be broken down in order to be transformed into glucose, which will then exit the cells and float around in circulation so that the cells can take up glucose in the starved state. So some endocrine responses to the fed state. So what happens once you've actually eaten a meal? How does the body know that it should release insulin? And so the first event that happens is you're going to have a high amount of nutrients in your duodenum, in your intestine. And so that'll signal to the intestinal endocrine cells that they should release GIP and GLP-1. So these are polypeptides that once they're released, that's going to signal to the pancreatic beta cells that, hey, you know what, we have a high plasma glucose. We should probably release some insulin in order to encourage the cells to take up that extra glucose. So then insulins will be released. It'll notify the liver tissues, which will then participate in glucose transport with glycogenesis, which is the production of glycogen. So once the glucose enters the liver in the fed state, the liver can then use that glucose in order to build glycogen as storage in case when you're hungry and you have low blood glucose levels, the liver can break down that glycogen to generate glucose. And then it can also build up macromolecules like lipids and proteins, also using that extra glucose. And there's one, sorry, Jose, one extra thing to mention here is that GLP-1, which is the polypeptide we talked about, will actually inhibit pancreatic alpha islet cells. And then also a high plasma glucose will inhibit the pancreatic alpha cells in order to discourage them from releasing glucagon, because like we said, we need insulin to dominate in the fed state. This is just a quick overview of what happens with insulin, its mechanism of action. So insulin will bind to a tyrosine kinase receptor. The receptor will then phosphorylate these insulin receptor substrates. The professor said that you don't need to know the exact name of them, just know that this event happens, they get phosphorylated. So that triggers a secondary messenger cascade that alters protein synthesis and the existing proteins within the cell. Membrane transport is modified, and then cell metabolism is changed, and we will talk about how that happens. So what's going to happen in the case of muscle and adipose cells? So the insulin signal will trigger exocytosis of GLUT4 to the plasma membrane. And so GLUT4 is this glucose transporter within the cell that is localized deep within the cell, but it won't go over to the cell membrane unless there's an insulin signal. So after you've eaten and insulin levels are rising, insulin will bind to the receptor, which will then encourage the GLUT4 receptors to move over to the cell membrane in order to take up all the extra glucose. So glucose in the bloodstream will enter the cell through facilitated diffusion, which if you can recall is a process that we discussed in the very beginning of the class. So glucose will enter the cell through facilitated diffusion. And then just one note to keep in mind is this process of exocytosis of GLUT4 to the plasma membrane only occurs in muscle and adipose tissue. It does not occur in the liver. So that's an important distinction to keep in mind. We will talk about what happens in the liver. So what happens at the liver? So in liver hepatocytes, we have GLUT2, not GLUT4. That's another important distinction. It could very well be a test question because it's tricky, 2, 4, but just keep in mind the liver uses GLUT2. And GLUT2 is always located on the plasma membrane no matter what, both the fed and fasted states. GLUT2 will always be located on the plasma membrane. It doesn't undergo exocytosis like muscle cells. So that's important for the liver to be able to do in order to mediate the immediate uptake of glucose, right? So if you think about it, the liver needs to know all the time what's going on with your sugar levels, right? Because it's such an important organ for metabolism. It should always kind of be in the know. It can't afford to, keep exocytosing its GLUT receptors or its GLUT molecules because it needs to know what the blood glucose levels are like. And so being always located on the plasma membrane, that allows for immediate facilitated diffusion of glucose in the event of high blood glucose levels. So right away, once insulin interacts with the receptor, GLUT2 will take up the extra glucose in the bloodstream, and that generates a cascade where glucose will flow from an area of high concentration from outside the cell into an area of low concentration. And if you're curious as to how that concentration gradient is mediated, well, if you recall, glucose is transported into, or it's converted into glucose 6-phosphate. phosphate, which is an event that occurs in glycolysis. And so inside the liver, you don't really have glucose, right? You have glucose 6-phosphate. And so that gradient of glucose high on the outside and low on the inside is pretty much constant throughout the entire time that you have a high concentration of glucose outside the liver. So insulin and glucagon blood levels. So with high blood glucose levels, like we said, there is a high insulin plasma concentration, and then glucagon levels are low. So these two hormones are always going to be antagonistic. So when one hormone is high, the other one is automatically low. You cannot have high glucagon and high insulin at the same time. And then vice versa, with low blood glucose levels, there is a high glucagon plasma concentration. So when you have a high glucagon concentration, that encourages these processes like glycogenolysis, gluconeogenesis, and ketogenesis. And these are all processes that your body is going to undergo in order to start up energy production in a low energy state. And then, like we said, insulin levels will be low if glucagon is high. So we talked about the endocrine response in the fed state. So what happens in the fasting state? Because it is quite a bit different. So in the fasting state, you're going to have low glucose levels in the plasma. Your pancreatic alpha cells are going to pick up on the low levels of glucose, and they're going to release glucagon. Glucagon will then be sensed by the liver, which will then undergo glycogenolysis or gluconeogenesis, both of which will increase plasma glucose levels. Now, keep in mind that your liver is the only thing that's going to be responsive to high glucagon levels. So your muscle and adipose and other cells are not going to sense the presence of glucagon because they're only sensitive to insulin. So there's going to be low insulin, like we said, in the presence of high glucagon. So then what happens, right? So muscle and adipose and other cells will sense that there's a low level of insulin. And so they will then start producing lactate, pyruvate, and amino acids. And these three substrates can be used in the process of gluconeogenesis and glycogenolysis. So again, remember what we said, many different substrates can feed into various biochemical pathways. And remember that fatty acids is the unique one, right? So fatty acids can't feed into gluconeogenesis or glycogenolysis. What happens instead is fatty acids will undergo beta oxidation to release ketones. And these ketones will then be used as an energy source for the brain and other tissues. An important fact to keep in mind is that the brain can only use glucose and ketones as an energy source. It cannot use fatty acids. It cannot use amino acids because they're just simply too large. It needs something small like glucose. So only glucose and ketones can be used to feed the brain. So let's talk about some plasma metabolic fuels under various hunger states. So when you are completely fed, you're not hungry at all, your glucose is going to be the main metabolic fuel. Okay, so we're not going to be using ketone bodies or anything like that because we have glucose, right? And that's sufficient for our metabolic needs. However, if you notice here, after 7 days of starvation, there's almost like pretty much the entire metabolic fuel is going to be from ketone bodies, right? Because we don't have any glucose that the cell can use in order to generate energy, right? So we're going to be relying on the process of fatty acids converting themselves to ketone bodies. So ketone bodies are, like I said, they're produced as a byproduct of beta oxidation. And again, like I said, they are the only source that the brain can use as fuel in the absence of glucose. And so they will readily be made in the event of starvation in order to keep feeding the brain and your other cells and tissues. So let's talk about type 1 and type 2 diabetes. So there are important distinctions between the two. So, however, for both conditions, cells are going to be unable to take up glucose. So that's the general theme, right? In diabetes, for some reason, your cells are not able to take up glucose. The reasons, however, are different, right? So for type 1, we're not going to be able to take up glucose because of destruction of insulin producing beta islet cells. And that's going to be through an autoimmune malfunction. So for some reason, our beta islet cells are destroyed. They're not able to produce insulin. And so the cells are not able to be responsive to the glucose that's in the bloodstream. So they're not able to take it up. That's different from type 2 diabetes. Type 2 diabetes is due to an insensitivity to insulin receptors. So basically, your insulin receptors are just not sensitive to insulin, essentially. So insulin can be there in the blood. In fact, it can even be elevated in the blood, but your receptors are not able to sense that. And so your cells cannot take up glucose. And then type 1 diabetes tends to be in very young individuals. It tends to be more hereditary, but type 2 diabetes, we think of it more as a lifestyle type disease. So what are some pathophysiological effects of diabetes? So one thing that we see happens is the cells will shift to a state of catabolism because of their inability to take up glucose. So the cells will actually start breaking down their own energy fuels because even though there's a lot of glucose in the bloodstream, they're not able to respond to it because of the lack of insulin or the lack of mechanisms that mediate proper insulin function. And so the blood can be hyperglycemic, meaning that there's way too much glucose. And in the case of type 2 diabetes, it can also be hyperinsulinemic. So that means that there's a lot of glucose and a lot of insulin. And then one thing that we often see as well is people will start to have polyphagia, which is an induced appetite that occurs because you keep eating and eating because your body cannot respond to the glucose levels that are already in your bloodstream. So you always feel like you're hungry because your cells are not able to use that food that you're eating for energy. Glucose tends to be excreted in the urine in very large amounts. So individuals with diabetes tend to have sweet urine. And that causes frequent urination because there's a lot of osmotic pressure on the kidneys to generate more water to get rid of the extra glucose in the urine. So you're going to keep urinating throughout the day, which will reduce blood volume, and that in turn has the effect of impeding circulation, right? So when you have reduced blood volume, you're going to have suboptimal circulation. So your blood is not going to be able to go into your extremities and your limbs, as well as a person that may not have diabetes, and that can often lead to very sad cases of limb amputation because of the lack of circulation to those areas. And sorry, Jose, one more. An inability to utilize glucose leads to reliance on ketone bodies, okay? So that's one extra thing to keep in mind. So you cannot use glucose that's already in your bloodstream because you can't take it up. So what's gonna happen? You're going to start producing ketone bodies, and then that's gonna result in metabolic acidosis. So that means that you're gonna have acidic blood, essentially. And so what happens from a biochemical perspective is our bicarb equation is going to be shifted to the left because we're going to keep generating these ketone bodies that are making our blood acidic, because like I said, we can't take up glucose, so we keep generating ketone bodies. And this is much more common in type 1 diabetes, just something to make note of. It can also be common in advanced cases of type 2 diabetes, but we generally see this mostly in type 1 diabetes patients. So I have a question here. Which of the following may be seen in a type 2 diabetic individual. This is going to be tricky to do the poll because there may be more than one right answer, but I guess just pick one that you think may be right. Okay, I guess we can end the poll now. You guys seem to get it. Okay. Yeah, so in type 2 diabetes, we would expect to see high levels of glucose as well as high levels of insulin. And then in some cases, we can also see high blood acidity due to ketone bodies. All right, lecture 22. Okay, so let's talk about temperature and heat. So these are not the same thing. That's a common misconception. So they are two different things and they measure two different things, right? So temperature is a measure of speed or intensity of molecules in constant random motion. So you're measuring their speed or their intensity, okay? And so substances that move faster will have a higher temperature, and substances that have a lower speed will have a lower temperature. So that's different than heat, right? So heat is the total energy that a substance possesses due to the random motion of these molecules. So the molecules that we talked about for temperature, heat is a measure of the energy of the motion of these substances. So how much energy are they generating when they are moving along one another? So heat will depend on both speed and the number of atoms. So just keep that in mind. So it depends on speed and number of atoms in the air. And so heat gain must equal heat loss in order to maintain a constant body temperature. So if you let out some heat energy, you're gonna want to equal that by gaining heat in order to maintain your body temperature. So you can think about that. You know, say you are out on the street, it's like mid-December and it's chilly, you're gonna put on a jacket, right? Because you want to achieve a state of homeostasis in your body temperature in order to make sure that the heat gain is equal to the heat loss. One other thing is the addition of heat to an object will also increase its temperature, which is kind of intuitive, right? So just one extra fact here about heat to keep in mind. So there are a few different processes for as to how heat can be transferred, right? So one is radiation, and radiation is the transfer of heat between two objects that are separated by air. And a very common example of this is sun radiation, right? Heat radiation. The sun can elicit warmth by radiating its rays. Another example or another form of heat transfer is conduction. And conduction is the transfer of heat through contact with a material that's not in motion. And so the part that's underlined here, not in motion, is important to keep in mind because that's the part that differentiates it from convection, right? So convection is different from conduction. It's the transfer of heat through contact with a material that is in motion. So an example of this would be the wind blowing on you, right? So the wind is moving, whereas with conduction, nothing is moving, right? So an example is like holding my phone, right? So the energy from my hands will transfer over to the phone. And then fourth and final process is evaporation. And I think we all know what evaporation is. So water will change from a state of liquid to a state of gas by absorbing a very large amount of energy. And it must reach 100 degrees Celsius in order for that to occur. Oh, sorry, Jose, I keep forgetting things. Just keep in mind also the equation on the right. This is an important equation for conduction. And so two important variables that determine conduction is range in temperature and distance or diameter. So the larger the range in temperature, the higher conduction. And then there's an inverse relationship between distance and conduction as well. It's distance, it's not diameter, I'm sorry. So an example or a question here is, which of the following is an example of conduction? A few more seconds. Okay, what was the right answer? Yes, so most of you got that one right. So yeah, keep in mind that conduction, like we said, is lack of motion, right? So cold pan placed on a stovetop, nothing is moving here, right? So this example of heat transfer is conduction. So let's talk about how the body is able to mediate such a constant body temperature. So if you look over to the top portion of this figure here, the kind of yellowish orange portion, so you can see that the body temperature is very, very constant, right? And so the lower range and the upper range are cases of hypothermia on the left and hyperthermia on the right, which are both extreme ends. But other than those two cases, the body is able to maintain very constant temperatures. And so when we do have these cases of very low temperature and very high temperature, we're actually exerting a lot of metabolic energy to keep our body's temperature maintained, right? So the thermal neutral zone is kind of the sweet spot where the range of ambient temperatures it's not so extreme to where the body can maintain a constant temperature without it having to expend too much energy, right? And so at these upper and lower extremes, the body is extending way more energy than it ideally wants to. Okay, so let's talk about the process of what happens when we have suboptimal temperatures. How does our body sense that? So the first point is we have these peripheral thermoreceptors and central thermoreceptors. So these receptors are able to sense a temperature change. And so these will then send that signal, whether it be a lower temperature or a higher temperature, to the hypothalamus, the hypothalamus thermoregulatory center. And so that hypothalamus will then integrate that signal, and then it'll initiate the appropriate change. So in cases where we have increased temperature, that's going to be controlled by sympathetic cholinergic neurons. So neurons that are mediated by acetylcholine, right? So that's an important distinction from decreased temperature, which we will talk about next. And decreased temperature are the neurons involved are adrenergic neurons, not cholinergic. But in any case, in increased temperature conditions, we're going to have sympathetic activation of these cholinergic neurons. So that'll trigger two cascades, either one of two cascades. So we're going to have these sweat glands release sweat, right? When we all know what that essentially is like. It's a very intuitive concept. You're hot, you get sweaty, and then the heat is then lost through evaporative cooling. And so like when you're working out, you're sweating, you want to cool down, your body does that by releasing this excess heat through the skin. And then one other mechanism is through cutaneous blood vessels, which will vasodilate. And vasodilation is essentially expansion of blood vessels, right? And so that'll then result in heat loss to the environment, right? So when you vasodilate, your blood vessels will let off any extra heat to the external environment. And then in contrast to that, we have decreased temperatures. And so a few different things can happen here. So like we said, we're going to have activation of the sympathetic adrenergic neurons, which will then activate the cutaneous blood vessels, but those will elicit a different response. So these cutaneous blood vessels will initiate vasoconstriction or constriction of your blood vessels, which will then conserve the heat to make sure none of that heat is worked off to the environment. You want to conserve as much of it as possible. And then another mechanism is through brown adipose tissue, which we will talk about in a few minutes. And brown adipose tissue can cause non-shivering thermogenesis. And we will talk about that more in detail when the slide comes. And then one additional mechanism that happens is through somatic motor neurons, which will act on skeletal muscles in order to tell us that we need to shiver. So shivering is essentially like very fast muscle contraction that generates warmth in the body. So brown adipose tissue, which was one of the means by which our bodies increase temperature, is different from normal white fat cells in that they have a much fewer number of lipid droplets, and the lipid droplets are also much smaller in size. The role of brown adipose tissue is essentially to convert chemical energy into heat energy in order to protect you against the cold. And the very unique thing about this brown adipose tissue is that it has a very large amount of mitochondria. And so this number of mitochondria essentially dictates its function, right? We want to have so much mitochondria in order to facilitate a very controlled amount of thermoregulation. And so brown adipose tissue has a very unique mechanism that is different than regular oxidative phosphorylation that occurs at the electron transport chain. So normally, under normal conditions, your protons that are being shuttled across the electron transport chain are going to be coupled to ATP in order to generate energy, right? So this is a bit different than what happens in brown adipose tissue. So brown adipose tissue generates this uncoupling protein that uncouples proton diffusion in the electron transport chain from ATP synthase. Okay, so normally we have protons coupled to ATP synthase in order to generate ATP, but that's not occurring in brown adipose tissue. Okay, so we have these two independent processes from one another. They're uncoupled. And so what the effect of that is, you have the energy of the concentration gradient of these protons is going to be lost to heat energy versus being used to generate ATP. And so that's how we're able to thermoregulate through this brown adipose tissues, because we're generating very large amounts of heat energy by not using the energy to make ATP, but instead using it to generate heat. And then this ultimately increases thermogenesis. So we have another question here. Which of the following is the method by which brown adipose tissue maintains thermoregulation? Right, I think you guys got it, so we can end the poll. Yes, good job. Decoupling ATP from protons. Okay, sorry, give me a second here. Okay, so let's talk about what happens during the fever response, which I'm sure is a response that all of us are familiar with. So your body temperature is kept at a very constant level, like we said. So about like 98.5, 98.6 degrees. So what happens when your body starts to fend off an infection is your body temperature will actually elevate to much higher than its normal set point is. And so what happens, what the body then does, the physiological response is that it'll generate these chills. You're going to start being really cold. You're going to start shivering. I'm sure all of you are familiar with what that feels like. But then once the body has kind of overcome the infection, once it has like fought the battle, so to speak, that temperature will start to immediately go down. And so how the body responds to that is you're going to start profuse sweating, right? So you just went from being super cold and you have chills and shivers to you're extremely hot and you're sweating. And so that's how the body responds to that immediate temperature change, is by vasodilation and profuse sweating in order to come back down to its normal steady temperature. All right, and thank you guys for listening. I'm gonna hand it off to Ariella.
Hi everyone. So I will be going over the first 3 lectures on kidneys and osmoregulation. So for lecture 23, this is mostly just anatomy and definitions. So if I've included some, but not all of what I thought was important. So to start off, kidneys. So the primary functions of the kidney are first, the removal of metabolic waste products and foreign substances, the regulation of plasma osmolarity, the regulation of plasma ionic composition, which includes sodium, potassium, calcium, magnesium, and all that good stuff. The regulation of plasma volume, pH regulation, and hormone production. Okay, so here's a picture of the urinary system. So the right kidney is responsible for the formation of urine and the regulation of the water balance in the body. And then these are the 3 definitions or 3 anatomies I thought were most important. The ureter, which transports urine from the kidneys to bladder, the urinary bladder, which is the reservoir that holds the urine, and the urethra, which is the transport urine out of the body. Okay, so here I compared the renal cortex versus the medulla. So the cortex contains all of Bowman's capsules and the proximal and distal tubules, and it's the outer layer of the kidney, whereas the medulla contains the loops of Henle and the collecting ducts, and this is the innermost layer of the kidney. And moving on, we have nephrons. So I would please remember what a nephron looks like in all parts. But this is the function.
I think Ariella might have frozen.
Did we lose
Oh,
Ariella?
no.
What happened? Where is she?
Ariella, can you hear us?
Yeah,
Ariella, can you hear us?
she said that her internet was going bad before we started.
Can you unmute yourself?
Hi, sorry about that. My internet dropped. Yeah, so to continue on. If you can move on to the next slide for me, Jose. Okay, thank you. So first off, there's cortical, and that composes 80% of the nephrons. So they contain short loops of Henle, and they have a slow rate of filtration. And their main function is excretory and regulatory functions. And on the next slide, we have the juxtamedullary. Remember, juxta means next to. So this is 20% of the nephrons, and it has a long, super duper long loop of Henle. And it has a high rate of filtration, and its main function is for concentrating or diluting urea. All right, so nephron function. So there are 4 processes of the kidney. So first off, there's filtration, where blood plasma and dissolved substances smaller than proteins get filtered into the glomerular capsule. And second, there's reabsorption, and this occurs all along the renal tubule and collecting duct, where water, ions, and other substances get reabsorbed from the tubule lumen into the capillaries and finally into the blood. The third step is secretion, where substances such as waste, drugs, and excess ions get secreted from the capillaries into the renal tubule and finally into the urine. Last, the excretion, and that's where substances are excreted from the lumen to the external environment. So GFF, or glomerular filtration. So this is driven by Starling forces, and what's the important part about this is the renal corpuscle, which is the site of fluid filtration, and it contains the glomerulus, which is the vascular element, and the Bowman's capsule, which is a tubular element. So the filter resembles plasma, except in lacking proteins present in plasma. And then moving on to the next slide, there are 3 barriers that you need to cross before entering. So there's this filtration barrier, which is semi-permeable, that allows for the passage of plasma and ions. So first, there's the capillary endothelial cell layer, and these cells have large pores that increase the movement of fluid through the cells by bulk flow. And then there's the layer, the basal lamina, and this is in between the endothelium and the podocyte layer. And these are just cells that are in between that also are semi-permeable. And then we have the podocytes, which are modified cells that create some filtration slits. And then it ranges in slit diaphragm, which is the final filtration barrier before fluid enters the glomerular space. And now we'll be moving on to GFR. And GFR is the volume of plasma filtered per unit time, which is approximately 125 milliliters per minute over the course of a day. So that means 180 liters is filtered. And the GFR is determined by pressure and the filtration coefficient. So this is highly regulated because the constant GFR is approximately 180 per day. This occurs when the mean arterial pressure is 80 to 180 milligrams of mercury. And so in addition, the filtered plasma is 99% reabsorbed and 1% is excreted from the body. And changes in the GFR have a large impact on how much can be reabsorbed, and that's why it needs to be so closely regulated. So for renal autoregulation, there are resistant changes in renal arteries, which alter renal blood flow and GFR. So this is what happens when there's increases in mean arterial pressure. So the key part is resistance in the afferent arteriole. And whereas in decrease in mean arterial pressure, there's a resistance in the efferent arteriole. So those are the main parts I would remember. So how is GFR regulated? So there's two forms of intrinsic control, which is myogenic regulation and glomerular feedback, and two forms of extrinsic control, neural and hormonal. And I thought this chart depicted all of them pretty well. So if you have time, I would go ahead and look at this for more detail. So the first one, myogenic autoregulation. So this is when, for example, the mean arterial pressure increases the pressure in the afferent arteriole, which causes a rise and stretch in the smooth muscle of the afferent arteriole. So this results in a reflex contraction of the muscle. And the vasoconstriction of the afferent arteriole causes this pressure in the capillaries to decrease. And whereas a decrease in mean arterial pressure would have the opposite effect. And moving on, the second intrinsic control is these words are so big and long, oh my gosh. Okay, feedback. So there's changes in GFR, which cause a change in tubular fluid flow. So this change in the flow is detected by the special cells of the macula densa. So these cells secrete paracrine that affect the contraction of muscles of the afferent arteriole. So for example, an increase in GFR increases tubular flow, which ultimately causes the afferent arteriole to constrict and reduce the flow, and vice versa. And moving on to the third one, the intrinsic mechanism, which is neural control. So distance and flow, which the local control usually drives the show unless there is an extreme systemic problem that's outside of the normal range. And remember, the normal range of mean arterial pressure was 80 to 180. So when there's an increase in norepinephrine, there's vasoconstriction, and when there's a decrease, it results in vasodilation. All right, so now I have a clicker question for you all. So the urinary system is composed of, so we're gonna start off easy to get y'all like into the system. Okay, I'm gonna do 5 more seconds. All right. So let's see, it looks like a majority of people know that it is B, awesome. Okay, so I have another clicker question. All right, I'm gonna give y'all maybe 10 more seconds because I'm feeling nice. All right. So majority says D, and the answer is D. Awesome. All right, so now we're moving on to the next lecture. So reabsorption. So reabsorption is the movement of filtered solutes and water from the lumen of the tubule back into the plasma. Oh, sorry, the poll is popping up. So the solutes must pass two barriers to be reabsorbed, the tubule epithelium and the capillary endothelium. And then in lecture, the professor focused on the two-step process of sodium active transport. So the key part is that first, there's simple diffusion that allows for sodium to pass through. And then the second step is active transport with the sodium potassium ATPase. And moving on to excretion. So when solute concentration is so high that all the carrier proteins and pumps are occupied, it reaches transport maximum. So when the plasma concentration of the solute rises to the point that the filtrate concentration exceeds the transport maximum, some of the solute starts to appear in the urine as spillover. And at this point, the renal threshold is said to have been reached. All right, so here I just did a short table comparing reabsorption to secretion. So they're pretty much the opposite, whereas the lumen's going to plasma, and then this is the solutes in water. And the purpose is essential ions, and more water are returned to the blood. And then whereas in secretion, molecules move from the plasma into the renal tubules to become part of the filtrate. So this process involves the same mechanisms as reabsorption, except in reverse once again. So substances that are secreted include potassium, hydrogen ions, chlorine, waste products. And the main point of this is to clean the blood. Okay, so for this, I would remember what these all are. So when they solute moving through the nephron, just filtration is equal to excretion. There's no net. When there's more filtration, there's net reabsorption. And when excretion is more, there's net secretion. And this formula, I would definitely recommend this in case they do a question. And on the next slide, I provided an image that just gives you an example. All right, and moving on to clearance. So clearance is just a way of measuring excretion rates. It's really useful for describing how the kidneys are handling one substance compared to another. So the relative clearance of solutes indicates how excretion affects the plasma concentration of one solute compared to another. For example, if the kidneys excrete potassium and sodium at the same rate, the excretion rates would be identical. So this is the formula down below. I would definitely recommend this. I feel like these are really common for them to ask a question on either clearance or excretion rate. So on the next slide, it's just this picture again, but with more in depth on the bottom, such as the clearance of insulin. And I would definitely recommend going over the math questions in class. That does take time to go over all of them, and just remember like the different examples, like in insulin, all that good stuff. All right, so I have a clicker question here for you. Urine is formed in a 3-step process. Which is the correct order? All right, so I'm gonna give y'all about 5 more seconds. All right. So majority says C, and the answer is C. Great job. All right, so I have one more question. So the primary mechanism for reabsorption of water in a kidney is All right, 3 more seconds. Okay, let's see. C, correct. All right, so now we're gonna be moving on to the last lecture. Okay, so urine is stored in the bladder until it's expelled in a process called myoduration. So the smooth muscle in the wall of the bladder is called the digestive muscle. So, and the inner smooth muscle at the neck of the bladder forms the internal urethral sphincter. So the flow of urine is also controlled by the skeletal muscle of the pelvic floor called the external urethral sphincter. So urine is delivered to the bladder on a continual basis. Oh, sorry, Jose, can you go back? Thank you. So the internal and external urethral sphincters are contracted, so urine does not leave the bladder and allows you to hold in your urine and the volume increases. So the relaxed detrusor muscles accommodate this expansion as well. And then increased activity of these stretch receptors activate parasympathetic neurons that cause the contractions of the muscle. And this also increases the pressure of urine within the bladder. So the stretch receptors also inhibit sympathetic neurons innervating the internal urethral sphincter and the somatic motor neurons innervating the external urethral sphincter. So this allows the bladder to empty. And then this reflex is also overridden by voluntary control, like when the bathroom, we need to use the bathroom. Yeah, and moving on. So I provided a picture of just common substances that leave and enter or are reabsorbed and secreted. All right, and then now we're gonna move on to water balance. So homeostasis requires water intake and output is balanced. So water balance is achieved in the body by ensuring that the amount of water consumed in food and drink and generated by metabolism equals the amount of water excreted. All right, so kidneys conserve water. So kidneys are the body's natural blood filter. So they're able to separate the reabsorption of water in solutes. And by controlling water, it stabilizes these fluid levels. For instance, if you're super dehydrated, they can make you more dilute to expel excess water if necessary. And FR, this once again, this is how much blood that passes per unit of time. And this is an important indicator of kidney function. And then, so the nephron. Throughout the nephron, there's osmolarity changes. And as you go down the medulla, it becomes more concentrated. And the countercurrent blood flow pattern leads to this osmotic gradient that helps drive water reabsorption. Okay, so for mechanism of vasopressin action, so the permeability to water generated by the insertion of aquaporins in cell membrane of nephron cells. So factors that stimulate vasopressin release are related to water conservation needs of the body. All right, and y'all can in your free time. So let's say, for instance, the body is conserving water. This is when vasopressin is released into the collecting duct epithelium, and then which makes it now permeable to water. So vasopressin is this hormone that's released by the posterior pituitary, and this controls the amount of water reabsorbed from the collecting ducts and tubules in the kidney. Okay, and so let's say, for instance, the body is getting rid of water, vasopressin is not released, which ultimately results in the collecting duct is not permeable to water. So due to the absence of vasopressin, urine will be what concentrated, more or less? And y'all can think about that since we're running low on time. All right, so for countercurrent heat exchange. So pretty much in the vasa recta, the main important parts is that this mechanism is done to preserve the osmotic gradient, and it acts as countercurrent exchangers. All right, and in the picture, it's a nice way to just look over how they, what factors play into maintaining the osmotic gradient. All right, so homeostatic responses to salt ingestion. So sodium intake raises osmolarity in the body, which triggers the conservation of water by the kidneys and stimulates intake of water, which makes you thirsty. So this is the pathway. So to restore ECF volume and blood pressure and osmolarity in the body to homeostatic levels. So this is a chart you can go through and just make up scenarios like, what if you ate too much Cheetos, or what if you only drink caffeine and no water, and see how it goes down the pathway. And moving on, we have the steroid aldosterone. So its primary function is for renal sodium reabsorption by the nephrons to regulate water and salt balance. So it makes the collecting tubule more permeable to sodium ions and increases water absorption by creating an osmotic pressure. All right, now I have a clicker question for you. So the renal system does not play a direct role in regulating what? All right, I'm gonna give 5 more seconds. Okay. The answer is B, correct. Awesome. Got another question for y'all. All right, 10 more seconds. All right. And the answer is B, correct. All right, so I have one more question for all of you.
You guys can go ahead and answer. I think Ariella might have frozen again, but just go ahead and put in your responses.
How convenient, huh? She freezes right at the end.
Perfect timing.
Talking about kidneys and urination, she probably had to go pee or something.
Mhm.
Okay.
She's back.
Oh my goodness. Sorry, I keep on.
You're totally fine.
All right, let's see what our answers is. We're ready? What do we do?
Most of you guys got it.
Did Ariella leave again?
Oh, I'm here. Okay.
Thank you guys for listening in. Thank you for coming tonight. We appreciate you coming out and hearing us. And feel free to look at the PowerPoint in your own time. We have like speaker notes in there and like other information at the bottom of the slides. So feel free to look at that on your own time. And we will be posting the recording later, either tonight or tomorrow. So be on the lookout for that as well.
Much. Best of luck to you all. You're gonna do great.
Yeah, you're gonna do awesome.
We believe in you. Stick around for questions if you have any.
Yeah, we'll be here for a few minutes for questions.
I'll stop the recording if I can remember how to do that.